My name is Luciano Bachmann. I'm co-author of this study. Here in this study we have the participation of my undergraduate student Carl Albino, my PhD student Luis Marcos Jr. Both all we are from Physics Department, Faculty of Philosophy and Science and Leather of Ribeirão Preto. For the same study we have the participation of researcher Juliana Faraoni and Professor Regina Palmadibi from Department of Restorative Dentistry, School of Dentistry, also from Ribeirão Preto. In this study, we measure the chemical composition depth profile of a carbon dioxide laser irradiated enamel. First of all, we must remember that carbon dioxide lasers has high affinity with phosphate radical. The phosphate radicals are in the inorganic matrix, and also the same wavelengths of carbon dioxide laser has high affinity with the water content from the enamel surface. So when these lasers interact with the enamel, produce high temperatures on the surface, but in the same time you don't have pulp damage, you don't have high temperatures in the pulp of the tooth. For this interaction, what you observe is a damage of organic material. The organic material enamel is very low, but you have this material is damaged. After that, you also have water removing. The water, if it is absorbed, is easy to remove, but in the enamel you have also structural water. It's water that are inside the inorganic matrix, and this water is more difficult. You need higher temperature values to remove this water. So finally, the carbonate radicals that you have inside the phosphate sites or hydroxyl sites from the hydroxyapatite, you can also remove with high temperature. So this laser, the carbon dioxide laser, can effectively change the chemical composition and also can change the structure of the teeth. For this research, we are employing only the wavelength 10.6, but remember that typical wavelengths for carbon dioxide laser are 9.3, 9.6, 10.3, and also 10.6, then the wavelength that we are using in this study. There is a large number of studies demonstrate the potential of these wavelengths to inhibit acid dissolution, to reduce artificial caries-like lesions, or also avoid dental erosions. Here we have 3 references, but there is a large number in the literature. Considering that there is a correspondence of the temperature rise on the surface and the carbonate loss in irradiated enamel, our motivation in the study was to measure how deep this change happens in enamel, in the irradiated enamel. And for this we can measure the carbonate loss, and then you can measure the deep profile of this carbonate loss after irradiating this enamel with a wavelength 10.6 micrometer. Measuring these changes of enamel of this tissue is possible to measure the deep profile first of the carbonate loss, and you optimize the laser parameters for desired application. Also to have a guess of how durable the enamel can be when you after a caries challenge. In resume, the objective of the present work is to determine the depth of modification, measuring the carbonate loss in an indenter enamel after radiating with a carbon dioxide laser with emission 10.6 micrometer. For methodology, we employ 25 bovine teeth and cut in blocks of 2.5 per 2.5 square millimeter blocks with a flat surface to irradiating. The irradiation was done with 2 different equipments of carbon dioxide lasers, both with the same emission. The beam power was gauged with a power meter infrared detector, and the beam diameter was measured with a knife edge method. Here are the parameters for the group G1, G2, and G3. Different number of samples. The first equipment, and here we have the second equipment. Different mean powers, pulse duration, mean radius, mean area, mean irradiance, pulse energy, and finally we have 3 different values for the fluence: 0.0180, 0.018 joules per square centimeter, 1 joule per square centimeter, and here higher fluence 3.7 joules per square centimeter. How we can measure the carbonate radicals? We employ a reflectance methodology attenuated total reflectance, name it ETR, coupled to a Fourier transform infrared spectrometer, so you have the absorption spectra over the 4000 to 700 wave numbers. Then you measure the absorbance, and you can measure the absorbance of phosphate, carbonate, also water content. To measure the carbonate, we measure some kind of an index. R is the ratio of the absorbance of carbonate and absorbance of phosphate. So this index is important to measure the relative content of carbonate to the phosphate radical. To access the information, the absorption spectra in the subsurface enamel layer, we do the following steps. First, we measure the absorption spectra of a non-irradiated sample. That is the index R, 0. After that, irradiate with a CO2 lasers, measure again the absorption spectra, and you have the index, the carbonate content, after laser radiation. So after that, you wear your sample to remove a first sub-micrometer layer of enamel. Then you measure again the absorption spectra, and then you can calculate the carbonate content for this sub-micrometer layer. Then you repeat the step 4 and 5 up to remove the entire change irradiated enamel. So we have accessed the absorption spectra of enamel at different subsurface values. To better explain the methodology, imagine that you have a block of enamel, here in blue. Then you irradiate your sample, you measure the absorption spectra, then you have this R, this carbonate content at the irradiated surface. Then you wear manually your sample and remove some micrometers, some layer of enamel. Then you measure your absorption spectra here. Then you remove again, measure the absorption spectra, remove again. So you remove layer by layer some kind of 1, 2, 3, 4 micrometers, and you have the absorption spectra for different steps in your enamel. The question is when you must stop in the enamel wear and FTIR measurement. Imagine that the first value of carbonate content is the value of non-irradiated enamel. After irradiating and removing step by step your enamel, at deep value of enamel you can measure again the carbonate content similar to the non-irradiated enamel. In this time you can stop to wear your sample. To exemplify one sample, imagine that you are calculating your R index. This is an index that measures the content of carbonate radical. Before irradiating, you have a higher content of carbonate. After irradiating, you have a lower content. After wear your first layer, you have a little higher content, but similar. You wear again, you have a higher value of carbonate content. Wear again, and you increase up to a value similar to the original value before irradiating the sample. Then you can measure again to be sure that you have the same value, and that is which you have up to 1.8 micrometers. The carbonate loss that you measure, that we present in the next slides, is some kind of a percentage value. Imagine that carbonate loss in this position here is 0, because these values are the same value observed before irradiation. How is possible to measure the layer that we wear with abrasive material? For that, we do some kind of indentation using a Microhard as indenter. This indenter has a length-depth ratio 7:1, and in this figure you can observe that if you have a length of 7 micrometers, you have a depth of 1 micrometer. The upper vision of the microscope is something like that. Then you measure in the microscope 7, and you calculate that you have 1 micrometer depth of this indentation. Before wear the sample, you have this information. You have, for example, a length of 7 micrometers. After wear manually the enamel for some minutes, what you can observe is a lower value of length. For example, 4. If you measure 4, that means that you have here 4 and the depth of x. If 7:1, 4 to x, can calculate how is the depth of this indentation. In this case, we have 0.6 microns. That means that if you are the first value 1 and now is 0.6, that means that after wear you remove 0.4 micrometers. In terms of the spectrum, that is the original spectra. After irradiating, you only reduce the carbonate content, and when you wear your sample, you have the irradiated surface here. After wear 0.3 micrometer, you increase your value, and if you remove more 0.3, then you have a higher value of carbonate content. How you can visualize the irradiated surface? Here we employ a 3D confocal laser microscope, and we have in this image the non-irradiated enamel. In the left side is possible to see the flat image, and here some kind of a 3D profile. For the three groups, we have here first in A, the irradiated with 0.0185 joules per square centimeter, here 1 joules per square centimeter, and here 3.7 joules per square centimeter. And here we have a high magnification for the surface radiated with 3.7 joules per square centimeter. In the first fluence, you have some kind of a mark from the laser beam. After that, you have some wave profile, and this profile is more present in the 3.7 joules per square centimeter. It's possible to observe also in this group and this group here some melted surface. For the first group, when we irradiate with 0.0185 joules per square centimeter, we can observe here for the samples the carbonate lost. Then after radiation we have a higher content of carbonate, and after wear the sample you reduce this carbonate up to a values to near 1 micrometer. Then you have the same value observed before radiation. That means that in this position, in this region, 1 micron beneath the radiated surface, the carbonate content is the same as non-irradiated sample. That means that the carbonate loss in this position is 0. Here in the second group, you have a higher carbonate loss in the non-irradiated surface. After wear the sample, the carbonate loss reduced to 0. That means at 1.6 micrometers, you have the same carbonate content as before irradiating with the laser. In the third group, you have a higher. You see you have here a percentage of 30 to 6% of carbonate loss. After wear the sample to different values, you have a carbonate loss near 0 when you reach 2.5 micrometers. It's important to know that these different values are from different samples. Then we have some variance from one sample to another, but the mean values represent the same value at the final depth when you observe the carbonate loss near 0. In conclusion, the employed laser, CO2 laser with emission of 10.6 micrometers can promote the carbonate loss, and with employed methodology it was possible to measure the depth profile in the enamel tissue. In resume, you have carbonate loss up to 1 micro after disfluence, 1.6 micro after 1 joules per square centimeter, and you can remove the carbonate loss up to 2.5 micrometers after irradiating with 3.7 joules per square centimeter. Thank you for the attention. I want to thank you also for the FAPESP, CNPq, and these projects, and for contact. That's it.